in 00, 3 p.m. And as far as Steffen is online, then Steffen, the stage is yours.
Can you please share this screen so that I can share my screen? Because one can only share one screen at a time. Can you see the screen?
Yep, perfect.
And it looks kind of decent. Okay, hello, my name is Steffen Brinkmann. I'm working at IEK2 at the Forschungszentrum Jülich, and I'm also part of the NFDI-MatWerk, where I'm in task area software and workflows together with Tilman Hickel. And today I want to introduce to you the Pasta ELN, which we developed because we really want to focus on experimental scientists. And we also believe that experimental scientists and numerical scientists have different needs. And therefore, this ELN is really focused on experimental scientists. And so the first problem that I personally see is that basically what we do if we want to use an online ELN is that we have to enter the files and the data twice. So we do double work. Once on the computer, I structure it in folders, put in perhaps an overview Excel file, and then I have to do the same in an online ELN. There's also quite some upload and download involved. So for example, if I want to edit a file, my origin file, then I download it. If I don't have it on my hard disk already, then I edit it and then I have to re-upload it. And then there are all these human mistakes that often happen that I simply forget to upload one data set, one image, and so on. So the goal is really how can we increase the ease of usage? And the solution here is that in Pasta, everything is at first local. So all the files are in your local folders, and also the database is local on your own computer. And then you press a button, and then everything is sent into the cloud. Since all the files are on your local hard disk or on your local computer, one can use the normal software, whether it's Origin, MTech, OEM software, ImageJ, whatever, right? Everything is on the hard disk. So how does it look in detail? We have on Windows, for example, the folder which is called My Documents, and inside there is a folder which is called Pasta Projects. And inside these projects, there's a single project, and then Pasta automatically creates a folder for the tasks and for the subtasks and so on. So let's go through that in detail. And what I have right here, you can perhaps see it, or hopefully you can see it. I make it a little bit, or I leave it like that, it's an empty database. So there's no project, no measurements, no samples. I have only have my two instruments, my GeoSEM and my tensile test. Nothing is inside. And now we want to create everything within this demonstration. So how does it work? Well, for projects, we have to add data. And since I don't want to type everything, I just copy it from the handout that we were given, the preparation, so copy. That's all I need, but I also can say that it's an active project. I can specify the objective, copy, paste. I can give comments about the methods that were used, and I can specify tags. I don't know, let's shout out DGM for organizing it so greatly. And then what happens now if I go into my folder, when I click the submit button, what happens in the background is that this folder is created, as you saw, and then it's also created in the database. And now I go into, for example, the database view or the folder view, and everything is there. Now let's create all the experiments that we want to do. I want to include all the experiments that were done. So we have some tensile tests where there were different batches, batch 1 and batch 6. Then we have some hardness measurements. We have some pore and the SEM data we have where they were again distributed in different batches. And we give this a little bit of a structure by changing the hierarchy level so that in the end we have a nice view like this. We have our tensile test with different batches, the hardness where we dump all the data in one folder, the pore analysis, the SEM fields. And now if I want to save this hierarchy, what happens, of course, on this side is that the folders are created, again, automatically. And within those folders, there are, of course, the subfolders. Right? So the idea really is within, like you saw it, like 2-5 minutes, we created the entire folder structure, and the same structure exists in the database as on our file system. I might mention somebody has his microphone on and we can hear some background noise. Okay, let's go back to the presentation. So, and after I've created all this information, I can just press a button, and that really literally looks like this button. I press this button and then all this information is sent to the cloud. And once the information is in the cloud, other team members, my supervisor, whoever can access this data, whether it's on a desktop or a cell phone. The second problem that I see is that there's a lot of manual data entry. So if we look for the example of an SEM, there we might have to ask for the magnification or the acceleration voltage, the working distance, and so on. And that takes a lot of time to enter the data. It also means that there are human errors. Sometimes I want to enter a 3, but by accident I type a 2. Even copy paste is not safe. I might copy the wrong part. And then there's laziness, right? I don't want to enter all this data, so I take shortcuts, and so then the database is only half full. And as noted already in some of the other presentations, I call it extractors. Extractors are really heavily used to get all the metadata from the files. This metadata includes the metadata that the vendor, so the company that produced the instrument, provides, like SEM magnification. But it also includes a thumbnail, which is basically a meaningful image that the scientist can use whether this experiment was successful or not. So it's not only a small image where you cannot see anything, but it's meaningful. And these recipes are small little Python scripts that the user can share and change. So this is the Python extractor, for example, for a CSV file. It's 10 lines of code, I would guess. You load the data. If you don't know Python, you can ask somebody who knows Python. You can share it, you can edit it. If you don't want to have it in red, you change this value to green, and then it becomes green. The user also, or the scientist, can identify their own data that he or she wants to collect from the data set, like the maximum value or the minimum value, and then the recipe. And that's all that is needed. And so the scientist can identify the own metadata that he or she thinks is important for the project. And these Python files are small and they can be shared just by email. So how does it look like in action? And in this case, we have two kinds of data sets. We got these Excel files, which are overviews, where the pore analysis was done and summarized, and where the tensile experiments were done and summarized. That's the first type of data that we got. And then we got the original data files that came from the instruments. So that is normally what I use on my system. So I carry it via a USB stick to my computer and then I use it. So let's do that. Let's see how that looks like. So here we have again our pasta. We go to samples. Right now there are no samples, but let's just import the Excel files that were there. So we go to the USB drive, to the pore analysis, and import the pore analysis summary. And what do we get? We get it in a nice tabular view. We get all the metadata extracted, like the number of voids in certain planes, the maximum pore size, and so on. We can, of course, also do that for the tensile test so that all the data is already included. And with the click of a few mouse buttons, we can really populate our database. So now what we have is the different samples, different batches. We can sort in any arbitrary form. We can look at the comments that were there like this. It is in the database. All the metadata is included. We have included all the data from these files. But then we also have this other data, these measurements that come from the real instruments. And there I do what I always do. I take my USB drive on one side, I take my view on the other side, and then I just copy paste. And since I want to, this didn't work. I copy paste the charge one or the batch one. And just to show you how fast it works, I just copy. That was the SEM, the hardness data. I copy the pore analysis cross sections. I copy. And lastly, I have to copy the tensor test so that you cannot say that I skipped the nice tensor test. So I copy them into batch one. And I do the same for batch 6. I know it took a while, but that's okay. So now I have copied all the data. And I don't want to enter anything by hand. I don't want to link anything by hand. I just want to go to my project. I go to the project and press one button, the scan button. And what happens now in the background is that Pasta scans for new files. And if it finds a file that was changed, it extracts the metadata. It generates a small little image that I can use to judge whether the measurement was successful. And then it goes to the next file and so on. So this takes a little bit of time because I included quite a number of measurements. And sometimes it apparently takes longer than other times. And now what you can see already is that many things were created here. The hardness measurements, well, they are not very interesting. They are just basically random measurements. Sometimes you see gradients in the hardness when you have a surface effects or something like that. You can have the pore analysis, seen these nice little squares that we've seen before. So all this data is in the database. But we can also look at the measurements now. And these measurements give us a more detailed view. Let's start with the hardness. Let's start with the most boring thing, and that was the hardness. It's plotted in line profile. I, as a scientist or user, have defined I also want to know the average hardness or the standard deviation, and those are shown. I have a link to the sample, so it's there. Again, I did not have to enter the link to the sample. It was automatically created because the sample information was in the file already. Why should I enter it? The next thing that is of interest is the tensile test. Again, the link to the instrument is there because it's in the file. The link to the sample is there, some other metadata. And now we have come to the recipes. Right now, or the default value is that I have a stress strain plot. But I can also say, no, I want to look as a function of force as a function of time. Tensile test is, in this case, displacement controlled. So force over time gives us a view of the response of the material. Interesting. Then I can look at the JPEG images, which are included, of course, with lots of metadata from the company of the microscope and so on, the resolution limit and so on. In this case, I have defined different recipes because different recipes make sense for these metallo images, which might be that I want to highlight the pores. And so right now I cannot see the pores. They are very small. And hopefully I didn't take the one image which does not have any pores. There's exactly one image that doesn't have pores. But let's see, at one of them I can highlight the pores. Here it should work. And there it is. So I can look, what is the distribution of pores? Are they all on one side? Do I have a gradient or whatsoever? But the best part is always SEM images. Those are the most beautiful. Like this one, you see nice raw SEM image with lots of metadata already extracted from where's the voltage. Sometimes I find the voltage fast, but yeah, I don't see it right now, but it's definitely in there. There's a voltage. It's in SEM. All this metadata was given by the instrument vendor already. And this default image is kind of old school. So I can say, I don't want this bottom bar, this data bar. I just want to have some meaningful domain. And then I say, yeah, what I also want to have really is a nice scale bar so that I can show it to my boss and she doesn't run away. And then I have a scale bar, right? And again, everything is linked, so I can automatically go to the instrument, can know more information about the operator or what instrument it was. Now, let's come to the third problem that I see is, and I as a group leader generally determine the structure of the data. So what data is important, what data should be saved. But sometimes I have to admit that scientists want to change that. They have a problem, they want to create a different structure, and then they would have to ask me as a group leader to change their data structure. Sometimes I personally just want to try something. I don't want to make a big change for the entire group. I just want to locally change something. And I also want that data structures change over time. And since everything is locally first, I have this flexibility. I can define totally different types of data, whether it's rooms. I personally like to work a lot with procedures. What is a different version of the procedure? What is the steps of the procedures? Each of these types of data can have totally different properties. Like a room would have square meters, perhaps corridor, room number, while collaborators would have name or email address. And there's only one requirement, and everything else is totally free. Everything has to have a name so that it can be displayed to the user. Luckily, or sadly, I don't have the time to show that today in detail. So let's just conclude. On which computers does Pasta work right now? It works on Linux. I've shown you that. It works similarly on Windows and on macOS. And we are also developing an app. Here is a screenshot from the Android App Store. And this app then allows us to read what is the procedure, what steps should I follow. It allows me to look at data, and it especially allows me to read QR codes. And these QR codes can then be on samples to track the samples. These QR codes can be on instruments to link instruments and samples much faster. And with that, I come to the conclusion where I introduced Pasta, where we really try to focus on experimental science. Everything, the database and the files are first local, and then one presses a button to synchronize. The extractors produce metadata. I did not enter any metadata by hand, you saw it. And they produce a thumbnail that gives the scientist a way to judge, is this measurement successful or was it unsuccessful? It's a flexible database which each scientist can totally modify as he or she pleases. And we are developing a cell phone app which scans the QR code and therefore even more simplifies the experimental research. And with that, I very much look forward to your questions and comments.
Stefan, thank you. Thank you very much. I very much appreciate your contribution. Maybe I can start with a remark about the boring hardness curve. And you as a material scientist understand. If you do any additive manufacturing, it's really interesting if you have a constant hardness over a section. That means that probably the material properties are homogeneous, and the scatter says something about the scatter, how it's scattered from powder particle to powder particle, and so on. You're a material scientist, you understand.
Yeah, and I didn't mean to degrade your research. I just sometimes we have these gradients, and
I did it not myself. And second is the question. did I understand correctly? Your highlighting of the pores and your nice scale bar thing, this was completely done within Pasta?
That the highlighting was, I mean, it's the same. I think it was a contribution already in from Humboldt University. They have to be, I call them extractors, other people call them parsers, whatever.
Okay.
And these parsers are little scripts that extract metadata or create the image.
Okay.
And in the case of the highlighting, for example, it's not so difficult. Where's the difference in gradient, color gradient large, put a red square around it. It's 2 lines of code. I don't have it right now in the presentation, so I cannot show you, but it's really not that hard. It's a small Python program that does the parsing or the extraction, however you want to call it.
As concerned pores, it's not so easy if you want to have a right value of porosity, because there's always there's a black pixel and a white pixel, and there's always a gray pixel in between. And how do you count it? But this is not the question here.
Exactly.
We discuss it when we meet next.
Yes.
Okay. Open to all. Stefan, do you have hands?
There are questions in the chat. 2 from Holger Israel. First one is about the cloud storage, what infrastructure is being used, and do users have encountered any data safety issue?
So cloud storage. The cloud storage is just a database Docker container. So you, just as in all the others, you have to install one Docker container in the cloud. It has nothing to do with Python. It's an off-the-shelf component which is called CouchDB. So it takes literally I would say 10 minutes to install. Then you have to give a username and password, and that's for the system administrator, and that's it. Data safety. As long as you don't click the share button or the synchronize button, all the data is on your local computer. So there's nothing with data safety. It's on your computer. People would have to hack your computer. Once it's in the cloud, it's of course, again, grouped into user rights. So the stuff that I produce and share with my supervisor cannot be read by a colleague and his or her supervisor. So that is the usual user rights. You can either read or you can read and write. And there are different groups for both of these. So yeah, nothing special there.
Mm-hmm. The second question is what makes Pasta more useful for experimental than for simulation workflows?
I mean, so in some past life, during my PhD, I've done a lot of simulations. And what I noticed is that in simulations, the data is often already in the cloud because the calculations are not done on my local laptop or computer, right? They are done on a server or a supercomputer. So once they are already on the computer, it is much easier to handle them also in the cloud. While in experiments, I often, and whether it's this institute or other institutes where I've worked at, you run around with USB sticks and you copy from a USB stick to your laptop to then process the data. That is not something that you do in simulation. So I think there are totally different workflows for both of these groups. And that's why we have written Pasta only for the idea of I come with a USB stick or I copy things from a central server onto my computer. I press one button, the scan button, all the data is automatically included.
Maybe this already answers the question of Christoph Pauli. Christoph, you can unmute yourself.
Can you hear me?
Yeah.
Okay, thank you. So if I want to get the data into my project, I first need to copy it to my local machine. I cannot access the project from like a computer in the lab or without making a local copy.
You can access it. My default example is that I access a picture on a Google server because it's easy to test whether the software is running. You can make a link to anywhere. Only the scanning does not work on, I cannot scan the entire network. That's simply not possible. It's also for security rights, right? So the scanning, the automatic scanning and the automatic taking of data into the database works only on your local computer. I can, of course, click here, measurements, add data, and then put the path in, or the link to the file on a Google server or on wherever. But yeah, so that's possible, but again, it involves a lot of steps, and I don't really see how relevant it is, because what happens if your instrument or instrument computer at a certain point dies because of old age? So these links to instrument computers are really not something that I see.
That's not the point, actually. I want to dump the data from the microscope computer into the project and then sync it down to my local machine.
Yeah,
so
that's
I do
of
not
course
rely
possible.
on the microscope. That's possible?
Yeah.
Okay.
Yeah, I mean, I right now search for files on my local hard disk. Why can't I search for changes of files on a hard disk somewhere else? That shouldn't be a problem.
Thanks.
Okay. And there's another question from Philip von Herod. Maybe you want to address it by yourself. Maybe it's not possible. And he wants to know if there's a possibility to use an example Omero for images for storage and analysis tools, in Klammern Fitchi. Also, is it possible to implement it?
Fitchi is ImageJ, right? Isn't that another name for ImageJ?
I'm not 100% sure, but I thought so as well, yeah.
So the files are on your computer. So I have right now not Fitchi or ImageJ installed, because I personally don't like to use it. Other people like it a lot. So yes, you can just use your normal computer stuff that you have on your local computer. I don't have to integrate it into the database, because everything is on your local hard disk or local computer anyhow. So there's no need for integration in my point of view.
Okay.
Yeah.
Stefan, I think we have to cut here and go to our, thank you, Stefan.